Great. I'm a little bit nervous, so I was going to tell you that and wait, get on with it. So I'm an ecologist by training, and I'm starting to dabble a bit in genetics. And today I want to tell you about how I applied population genetics to the traditional surveys that I did to answer some more questions about my sample site. I guess I need this too. So as background, brook trout are one of the few native stream salmonids in the Northeast, and they're facing decline across the native range, which is from the eastern provinces of Canada south to Georgia. Their decline is attributed to land use change over time and habitat fragmentation, which has also subsequently led to higher water temperatures. And there's also a long history of stocking in the Northeast to restore the fishery and enhance and supplement a recreational fishery. Increasingly, conservation genetics is emerging as an effective way to understand the health and genetic status of a fish stock and to understand the regional relatedness of populations and the effectiveness of restoration techniques and rehabilitation efforts. You can also use it to understand the impact of historical management techniques, like stocking. So there's thousands of miles of cold water, small streams in New York State that contain wild brook trout. And these are concentrated in the Catskills, the Tug Hill, and Adirondacks especially. The Adirondacks is a 6 million acre park made up of state and private land in the northern part of the park. And it contains lots of protected, forested, and connected habitat for brook trout. My study focuses on the Ausable watershed in the green here in the northeastern part of the park. This is a 512 square mile watershed. The river is 90 miles long, and there's two branches that originate in the High Peaks Wilderness and flow out to meet in Ausable Forks and flow in a northerly direction to drain into Lake Champlain. Both branches are fed by more than 70 cold water tributaries, and these and the protected headwaters offer excellent brook trout habitat. My study focuses in the Ausable Lakes in the headwaters of the east branch of the river. Located entirely on private land at the Ausable Club and the Adirondack Mountain Reserve. This was created in 1887. It's a 7,000 acre preserve with extensive access to hikers, but only the members can fish in the waters. The habitats are intact and almost pristine since it was created, and there's a native species assemblage in the upper lake. On the property, there's two lakes, Lower Ausable Lake and Upper Ausable Lake, and then there's 21 streams, the majority of which have brook trout. And I was brought on by the Ausable Club to help answer some questions they had about the status of the brook trout population. My initial research questions were whether and where natural reproduction was occurring in the watershed, how fish use the streams, where they are present throughout the watershed, and whether the hatchery fish are reproducing or if they're surviving in the system. So the lake is managed. It's been stocked since the 1980s with brook trout, and all the fish that were put in got unique fin clips. But the fin-clipped fish were not being seen in the fish surveys that took place from 2010 to 2015. Only a few, 3 fish. And then the creel surveys done by the members where they were educated about the fin clips and asked to report them, only reported 10 fish over the same time period. So that led us to believe that there was a wild fish population, and we suggested that the club stop stocking in 2015. So stocking was halted. In order to try to answer where the hatchery fish are going, you can look at whether, so it's documented that hatchery strains can have a lower diversity and reduced fitness in the watersheds that they're put into, and there's also hybridization that is well documented between wild and stocked strains, which over time can lead to a change in genetic population structure when they're mixed. But on the other hand, there's studies that show that there's minimal impacts on wild genetics by stocking, including in a study done at Elk Lake right over the mountain in a similar system. So based on my findings and the traditional surveys and those that were done before me, I hypothesized that there's a self-sustaining population in the lake, mostly wild fish, and that the lake and the tributaries that flow into the lake the populations would be related, and the fish would use the streams for spawning and nursery habitat. And also that there would not be a big hatchery impact on the population. And I think that perhaps the hatchery fish were facing natural mortality, angling mortality, or leaving the system in some other way. We're able to use a published set of 13 microsatellite loci, which are unique markers in the gene sequence of brook trout that were identified to help assign multi-locus genotypes and look at their historical demographics. From here, we can cluster them into inferred populations and determine whether the fish that I caught were unique in different parts of the watershed, like the tributaries, whether they're related, and whether there's a hatchery influence. You can do that by comparing genetics from the historically stocked strains. So I obtained fin clips through backpack electrofishing surveys and trap net surveys on the property. I also got a few hatchery fin clip donated to me. And then in the lab at the New York State Museum and University of Albany, I was working with Spencer Bruce, who's doing a similar project across New York State. And he showed me the ropes and got me going on to do all the DNA extraction in the lab. And then we used primers to amplify the 13 loci using PCR and a genetic analyzer to help us develop the genotypes. From there, you can go into a number of downstream analysis software to work up the data and infer what populations you have in a program called structure. So these are the streams where I collected the fish from. We got 52 stream fish from 3 streams that flow directly into the lake, and then Sawtooth Brook flows into the river that's downstream from the lake. We got an additional 69 fish from the upper Ausable Lake in trap net surveys that took place in May and October of 2016. And Spencer donated a population of fish from Casey Brook, which is at the top of the watershed. It's off the club property, and it's separated from the lake by a waterfall, but it's in the same, the stream flows right into the upper lake. SUNY Morrisville donated the 35 fin clips for our study. We had all the other hatchery strains to use for comparison, but except for SUNY Morrisville, which was stocked in 2010 and 2011 in the upper Ausable Lake. We also included populations from heritage strains in New York, but I'll not be presenting data on that today. So, with structure program, we did multiple model simulations using the genotypes that we created to split the sample into populations. And FST is a measure of population differentiation due to genetic structure, and our values in our Ausable sampling sites were very low and non-significant, which suggested that we had panmixis in the population. So we had a single free-flowing population between the lake and the streams, including the downstream stream. And so here are our major results. On the far left, you've got the stock strains, and on the right side, we've got the wild-caught fish that I caught in the system. And this is a stacked bar, and each vertical bar represents a single fish. And so the colors correspond to the genetic strains that you see within each fish based on the structure results and the genotypes that we created. So here on the far left, you've got the domestic strain in orange, and then the Temiscamese in purple, and then so the hybrids become a sort of half and half to help explain what we're seeing here. The SUNY Morrisville strain is unique from these others. They were actually sourced from Little Tupper Lake, and they've been raised in the hatchery for more than 18 years since they got the source stock. And then on the right, you've got the wild-caught fish. So the blue represents this Ausable Lake sort of native genetics, and then you see multiple introgression of these other colors from the left side. So you see a lot of domestic and Temiscamese genetics coming into these fish. The interesting thing is that Casey Brook here does not have very much, if at all, introgression from the hatchery strains. And you see very little, except for maybe Sawtooth, you see very little SUNY Morrisville strain in the fish. This is a similar plot that shows a similar thing. It's a PCA discriminant analysis of principal components, comparing all of our study samples. So the numbers here are inferred genetic clusters corresponding to each population, which are labeled on the left. And on this axis, you see, the domestic fish are on the top and the Temiskaming are on the bottom, separated from each other, but the hybrids fall in the middle of the two. And then our upper Ausable lake fish are over on this other axis between the hybrids and this Casey Lake, or Casey Brook strain over here, which is separate and not mixing. This is one other way to put it, and so what we've got is not a definitively unique population here. We've got and not much population structure for such a large system, which is surprising to us. You've got the fish in the lake that are just doing their thing, the resident fish, and then you've added hatchery strain fish. They're mixing with those. You've added other strains like the Morrisville strain that is in the lake for a period of time but is not showing up in the genetics, so it's dropping out in one way or another or leaving the system, possibly moving downstream. And then you've got lots of mixing between the lake and the tributaries, back and forth. And finally, the Casey Brook fish are isolated from this other population and moving around. So the population structure between sample sites is very weak to non-existent, and we can conclude that Upper Ausable Lake has a mostly admixed population of native and Temiskaming hybrids, parentage. Our measures of effective population size, FIS, and tests for heterozygote deficiency all suggested that the population's in decline. It's contracting or bottlenecking and there's likely inbreeding occurring. That was the most surprising result that we had. But Casey Brook, on the other hand, does not show the heterozygote deficiency. So this population is strong and it's growing. So the original strain of fish in the lake might have been the same. One possibility to explain this is that the extensive stocking in this system has caused the admixture across the watershed that's potentially catalyzing the decline of the population. The messages that I plan to take back to the club is, at first, to continue to not stock. The halt in stocking was probably a good idea, and until we can do some more research to figure out what can help restore the population, it'd be good to not stock. It's also possible, and it's been documented, that wild fisheries and genetics will recover if the stocking influence is taken away. And with these unexpected results, we need more research on what can be done. And you could possibly put limits to angling, like size limits to release pressure from the larger fish in the system is a possibility. And then I'd like to also consider the possibility of getting brood stock from this Casey Brook and some of the other isolated streams in the watershed, as long as it didn't impact the size and health of those populations, to think about trying to breed those fish to help try to rescue and restore the population. I'd like to acknowledge the New York State Museum and the University of Albany for extensive help and time that they spent with me to work on this project, especially Spencer Bruce and Karen Preston in the lab, the Ausable Club staff and the fishing committee, as well as advice and previous research from the folks that worked at the club, including Dan Josephson and Margaret Murphy, as well as my field assistants. Here's some of my funding. If there's any time,